Welcome to the Induced Innate Immune Response module. In this talk, we're going to learn about the different mechanisms that are used by host innate immune cells to kill microorganisms. We're going to begin looking at different types of innate immune cells and their functions. We will then look at how microorganisms and their components are taken up by innate immune cells and the different mechanisms that the innate immune cells employ to kill pathogens. We will finally look at examples that pathogens use to evade the innate immune cells. Our body has many different types of innate immune cells that play important roles in host defense. Some of them are the granulocytes, which include the neutrophils, basophils, and eosinophils. We also have monocytes that are normally found in the blood, and macrophages that are found in tissues. We have mast cells that are present in peripheral organs, as well as innate lymphoid cells that also play important roles in host defense. Finally, we have dendritic cells, which are cells that act as a bridge between innate and adaptive immune responses. Let us now revise the different phagocytes that can ingest and kill pathogens. Now, as we learned in the past, microorganisms are able to colonize the host because they're able to overcome epithelial barriers and invade tissues. And when that happens, they can encounter phagocytes or some innate immune cells. The main classes of innate phagocytic cells are the macrophages, monocytes, granulocytes, and dendritic cells. We looked at all of these populations in the first chapter, so we're going to revise them in the next set of slides. Now, to kill a microorganism, the innate immune cells have to find the microorganisms and they have to migrate towards the microorganism. And as we saw in previous part of chapter 3, this process is mediated by chemotaxis. So chemokines are produced at the site of infection, which allow the innate immune cells to know where to go to find the microorganisms. Now, after finding the microorganisms, the cells can now phagocytose the microorganisms to form the phagosomes. Once the organism is in the phagosome, the innate immune cell has different mechanisms to kill the pathogen, and we're going to look at 3 different mechanisms. The first one is a respiratory burst. The next one is the fusion of the phagosome to the lysosome, which we already touched upon a bit in the beginning. And we're going to look at a process called etosis. Before we learn about the killing mechanisms, let us first look at the cell signaling mechanisms involved. So, antimicrobial killing can be stimulated by cell surface receptors present on the host cell, and most of them belong to the family of G protein-coupled receptors or GPCRs. These are transmembrane proteins with 7 membrane-spanning alpha helices, and here we can see another figure where we can see the actual alpha helices, and the red is basically where the ligand binds to the G protein-coupled receptor. These receptors help immune cells move towards microbes and increases their antimicrobial activity through the process of signal transduction. And so it activates different signal transduction cascades that help in the immune cells to move towards microbes. Some examples of receptors that aid in microbial killing are the F-met-Lu-Phe or FMLF receptor. F-met-Lu-Phe is a peptide that is made by a lot of microorganisms and hence can be sensed and recognized by the FMLF receptor. The C5a receptor that recognizes the anaphylatoxin C5a is also a G protein coupled receptor that can help in the innate immune cells becoming better at killing the microorganism and increasing their phagocytic ability. So as we saw, RAC and Rho can be activated by G protein signaling, and RAC and Rho are members of the RAS family, and hence they are monomeric GTPases. They play important roles in actin organization and hence can regulate the movement of a cell. RAC activation promotes the formation of lamellipodia, which is a sort of a cytoplasmic structure or protrusion that is formed on the surface of the cells. Rho activation, on the other hand, promotes the formation of stress fibers. The activation of RAC and Rho helps the host to migrate in a specific direction. And that is what chemotaxis is, which is movement of the cell towards or away a diffusible chemical. Thus, RAC and Rho help in the cell to move towards the bacterium.
Neutrophils are white blood cells that hunt and kill bacteria. In this spread, a neutrophil is seen in the midst of red blood cells. Staphylococcus aureus bacteria have been added. The small clump of bacteria release a chemoattractant that is sensed by the neutrophil. The neutrophil becomes polarized and starts chasing the bacteria. The bacteria, bounced around by thermal energy, move in a random path, seeming to avoid their predator. Eventually, the neutrophil catches up with the bacteria and engulfs them by phagocytosis.
Let us now go through the different mechanisms by which the innate immune cells are able to ingest microorganisms and their components. So the first mechanism we're going to look at is phagocytosis. So in phagocytosis, it involves the uptake of solid particulate particles. The next one is macropinocytosis, and this involves the ingestion of fluid. So whatever is being taken into the cell is not particulate in nature, but usually soluble in nature. The final one we're going to look at is receptor-mediated endocytosis. And this is a mechanism of uptake of specific particles using receptors that are present on the surface of the cell. Let us now first look at phagocytosis. Phagocytosis is initiated when receptors on the phagocytes interact with molecules on the microbial surface. Now, if you look at the picture, there are many different receptors that are shown, and some of them, like the mannose receptor and dectin-1, are able to bind to carbohydrate moieties present on microbial surfaces that facilitate the uptake of these microorganisms. We can also have receptors like scavenger receptors and lipid receptors that can bind to anionic polymers and acetylated lipoproteins present on the microbial surface. We also have complement receptors that can bind to the complement proteins present on microbial surfaces. And finally, there is the Fc class of receptors, and these receptors can bind to the Fc regions of immunoglobulins. We will learn more about immunoglobulins when we start learning about adaptive immunity. In the process of phagocytosis, the microbe interacts with the cell surface receptors on the host cell, and this promotes the internalization of the microorganisms. And that is what we are seeing in that black box on the figure, where the bacteria and the yeast are interacting with the cell surface receptors, and that is promoting their internalization. The microbes get internalized into the phagocyte in a vesicle that is called as the phagosome. The phagosome is not shown in this figure. However, the phagosome can fuse with the lysosome to form a phagolysosome, and that is what is shown in the figure in the black box. The lysosome has many hydrolytic enzymes and antimicrobial peptides, and so by fusing with the phagosome, now all these enzymes and antimicrobial peptides can act on the microorganism, and thus the microorganism can be killed in the phagolysosome. The next mechanism of uptake that we're going to look at is receptor-mediated endocytosis. In this case, if we look at the figure in the black box, we can see that there are cell surface receptors on the host, and they are interacting with soluble ligands, which are shown as those blue or greenish dots that you can see. So once we have binding of that ligand to the cell surface receptor, a vesicle is formed, And this vesicle fuses with endosomes, and ultimately it will fuse with the lysosome. Now, during this process, as shown in the figure in that black box, you can have a dissociation of the ligand from the receptor, and depending on what kind of a receptor it is, the receptor can get recycled. However, the ligand is always sent to the lysosome where it gets degraded. If the receptor is also going to be degraded, then you don't need to have the dissociation of the ligand with the receptor. However, if the receptor is to be recycled back to the plasma membrane, it is kept in a different vesicle, and that vesicle goes back to the cell membrane. The next form of uptake that we're going to look at is macropinocytosis. In the case of macropinocytosis, as we can see here in the black box, the cell gives out a cytoplasmic protrusion, which is formed due to the arrangement of the cytoskeleton. And as we see the finger-like projection coming from the plasma membrane, it then engulfs the surrounding fluid to form an endosome, as shown in that blue box. That endosome will ultimately fuse with other endosomes and to the lysosome, and whatever contents were present in that endosome will be processed by the enzymes present in the lysosome. Now that we've looked at migration, let us now see what other things RAC activation can do. Now, RAC activation can also increase the antimicrobial activity of the innate immune cells by producing reactive oxygen species in those cells. For this purpose, we're going to look at the enzyme called NADPH oxidase, which is a multi-subunit enzyme that is usually inactive in unstimulated immune cells. NADPH oxidase can be activated when RAC is active. What NADPH oxidase does is it utilizes NADPH and oxygen, and it catalyzes a reaction that ultimately results in the production of a superoxide radical. These superoxide radicals are highly reactive and can act on the microorganisms. Thus, the NADPH oxidase activation, it causes a transient increase in oxygen consumption. So when it was discovered, people noticed that these activated innate immune cells are increasing their oxygen intake, but at that time they didn't know for what reason, and they just thought that it was an increase in respiration, and hence it was called as a respiratory burst. However, now we know that it's not merely for a respiration purpose, but that increase in oxygen consumption is for the production of these superoxide radicals. Let us now look at the NADPH oxidase. Now, it is a multi-subunit enzyme, so it's made up of many subunits, and I've listed all of them here. However, you don't have to remember all these names. So, in the cytosol, which is towards the lower part of the phospholipid bilayer, we can see different components, and just to know where the phagosome part is it is on the upper side of the phospholipid bilayer that we can see. Okay, so now let us look at in the unstimulated state what happens. So in the unstimulated state, we have the different subunits of NADPH oxidase like NOX2 and P22-FOX, and both of these, as we can see, are present on the membrane. The other subunits, which are the P40-FOX, the P47-FOX, and the P67-FOX, are all present on the cytosolic side. However, they're not associated with the NOX2 and the P22-FOX in the unstimulated state. However, in the activated state, this is when RAC gets activated, and RAC in its GTP bound state is able to associate with NOX2, and in that it also facilitates the interaction of the other subunits to the NOX2 and P22-FOX. And this way we now have the intact NADPH oxidase, which can catalyze the reaction. So it uses NADPH and oxidizes it to NADP+ by taking oxygen, and one of the byproducts is going to be that superoxide radical. As you can see in this figure, that superoxide radical is not made in the cytosol. It is made in the phagosome, and that is where your microorganism or pathogen is. Hence, we can see superoxide radicals being made in the phagosome in higher concentrations. The superoxide anions that are produced by NADPH oxidase are very reactive and can be converted to other reactive molecules as well. So when we look at the graph that's shown here we have our superoxide anions that are produced by the NADPH oxidase. In this figure, it's also called as the phagoside oxidase. Now, these superoxide anions can be used to make hydrogen peroxide. They can also be used to make other kinds of reactive molecules like the hydroxyl radical or even hypochlorite ions. And this way, by forming all these different types of reactive molecules that include the reactive oxygen species, it enables the phagocyte to kill microorganisms. Let us now look at the role of certain primary and secondary granules that are present in neutrophils. So as you know, neutrophils are granulocytes, and they're called granulocytes because in the cytoplasm we see granules. So in neutrophils we have the primary granules and we have secondary granules. And these granules contain many antimicrobial peptides like defensins and catalysidins. And we had learned how defensins and catalysidins work in terms of destroying or killing microorganisms. Now these granules along with the lysosome can fuse with the phagosome, and that way the defensins and catalysidins and other antimicrobial molecules present in these granules can act on the microorganisms. Now let us try to put all that we learned together and see what's happening in the slide. So let us imagine that there is a bacterium in the vicinity or at a certain site in the body, and it is producing this tripeptide, which is the FMLF. And we learned that FMLF can be recognized by a G protein coupled receptor that is present on the neutrophils. And with that, we will activate the G protein signaling cascade, and RAC2 gets activated. RAC2 is now going to start rearranging the cytoskeleton of the cell, and so the cell will start migrating towards that site of infection. At that site of infection, it will find the bacteria, and it will bind to it based on different receptors that are present, and eventually this bacteria is going to be internalized into a phagosome. Now, once this phagosome is formed, other parts of the cell will come to fuse with this phagosome. And these include the lysosome, the primary granules, and the secondary granules. And all three of these are shown in black boxes in the figure. So once the lysosome and the secondary granule and the primary granule all fuse with that phagosome, we now end up with the phagolysosome, which has the bacteria in it. Now, when we look at this specific figure, we can see that we have the NADPH oxidase assembled because remember RAC2 is active. So RAC can allow all the other subunits that are in the cytosol to assemble to that NOX2 and the P27-FOX. And we now have a functional NADPH oxidase that is making a lot of reactive oxygen species inside that phagolysosome, which will act on that microorganism. Those superoxide anions that are formed can also form hydrogen peroxide, they can also form hypochlorite ions, and other kinds of reactive oxygen species. Another thing to look at and pay attention to is now we can see, and we didn't talk about this that much, but we can see that there is a potassium channel that has become activated, and so there is an influx of potassium ions into the phagolysosome. Now what that means is in the cytoplasm there is a loss of potassium ions. And if you remember your NLR family, that change in potassium ion concentration is a trigger for NLRP3 to become active and start the formation of the inflammasome that will result in the cytokines IL-18 and IL-1 beta. they will be secreted in the environment and they can act on other immune cells and other parts of the immune system. Thus, by having all these different granules and the lysosome fusing and having reactive oxygen species being made, we can have the effective destruction of that pathogen. Let us now look at a recently discovered phenomenon called extracellular traps. Now for that, let's pay attention to this figure and look at the different components that are shown in this figure. So in this figure, we are looking at the neutrophils, and there are 3 neutrophils that are shown in yellow. Along with these neutrophils, we have some bacteria, and they are shown as these orangish rod-like structures. Now, what we see in that irregularly sized shape are blue-colored structures. These are extracellular traps. So immune cells like neutrophils, macrophages, mast cells, etc., they can form these extracellular traps that are used to target and trap extracellular pathogens. These traps are actually made up of nucleic acids. The process of making these new extracellular traps is called etosis. Etosis is a type of cell death, and during the process of the cell death, it involves the production of these net-like structures, or these extracellular traps. Now in this figure, we're looking at a neutrophil. Now neutrophils were the first immune cells where this phenomenon was discovered, and that's why it used to be called as netosis, or neutrophil extracellular trap osis. The osis is because this is a form of a cell death, but it's not apoptosis, nor is it necrosis. It's a different type of cell death. Now we know that in addition to neutrophils, many other cells can also show this phenomenon. Now going back to the figure, we have a neutrophil, and what can happen is a net can be formed in such a way that the DNA is condensed and the chromatin becomes really thick. And when that happens, we can see the production of a net. And eventually this net is going to rupture the plasma membrane and come out of the cell. And when that happens, we have formed the extracellular trap that can trap all kinds of organisms. Another way etosis can happen is where you form an extracellular trap, but its assembly normally happens outside the cell. In this case, the cell does not lose its plasma membrane or disrupt or rupture its plasma membrane. However, it exudes out its chromatin to form the extracellular trap. In both cases, the formation of that extracellular trap will result into the death of the cell. However, the extracellular trap will be able to catch all microorganisms that are there in the vicinity, and thus it allows other immune cells to efficiently find them and clear them. With this, we've looked at many different mechanisms that our immune cells will utilize to kill pathogens. And now we're going to look at how do the pathogens fight back. So how do they try to evade the immune cells and survive? So one way of doing that is that they prevent phagocytosis to happen. So for the neutrophils or the monocytes to effectively kill the pathogen, that phagocytosis step is an important step. By preventing phagocytosis, the cell or the pathogen is able to escape death. It usually does that by making a capsule. Now, phagocytic cells do not have receptors to recognize a capsule because most of the times these capsules are made up of sugars. However, the complement can still deposit on these capsular surfaces, and that can trigger the process of phagocytosis. So what I'm trying to say is when a microorganism has a capsule, the innate immune cells aren't able to latch on to the capsule directly. They need help from other systems like the complement or the antibodies in order to phagocytose the organism. Another way pathogens are able to escape death is by preventing the acidification and fusion of lysosomes with the phagosome. The actual destruction of the organism occurs only when the lysosomes fuse with the phagosome, because they are the ones that have all the enzymes that will be acting on the organism. So by preventing the acidification and the fusion of the lysosomes, many pathogens are able to escape death in that phagosome. This is shown by different strains of mycobacterium that can cause tuberculosis. Another way that pathogens are able to evade phagocytosis is they have evolved mechanisms to escape the phagosomes. So they do get phagocytosed, however, when they're in the phagosome, they have a way to escape from the phagosome and enter the cytosol of the cell. This is observed in Listeria monocytogenes, and this organism is an intracellular pathogen. So it gets efficiently phagocytosed by the immune cells, however, it has a way to escape from the phagosome and enter the cytosol where it's able to happily grow.
With this, we come to the end of our talk where we learned about different innate immune cells and their functions. We also learned about how host immune cells are able to employ mechanisms like phagocytosis and other endocytic mechanisms to take up microorganisms and their components. We also learned about different mechanisms that cells employ to kill bacteria, as well as different mechanisms used by pathogens to evade death by these innate immune cells.